here with Hans-Peter Fuluan. He is the lead scientist, genetics, for cucumber with bare vegetable seeds. Thanks for being with us today, Hans.
Oh, thank you for the invitation.
Tell us a little bit about your work.
Yeah, my work is basically, I'm scientist genetics. I give support to breeders. In the past, breeding, as breeders would call it, is an art. They selected plants. Nowadays, technology has catched up. We have lots of molecular information. We know the whole genome sequence of cucumber, and so now we can try to find correlations between genetic regions, and we call that markers, and the trait of a breeder. And once I know that, I can select in a very early stage already for plants that have the right combination of traits. And this will definitely speed up the breeding process. So basically, I'm trying to integrate molecular information into the traditional breeding process. That's my duty.
And you use AgriSeq targeted GBS. Can you tell us a little bit about how that was used in your most recent project?
Yes, we stumbled upon AgriSeq technology because we were using markers, but at a low density. So you have a big genome, and then if you have only a limited density, then yeah, you find a region, but it's huge. If you have a higher density, no, then the region is much smaller. So we want to use more markers, and AgriSeq offered that possibility. It was cost effective, and it has a fast turnover time, which basically means that, whoa, within a week I will have my results and I can help the breeders immediately to select their plants. So that's why we went with the AgriSeq technology in a pilot project. We choose the technique and we are now trying it.
And how did it compare with some of the previous methods that you've used in the past?
Yeah, like the previous method was fine, but the density was not high enough and it was expensive. So we wanted a cheaper method that was fast and that had many markers. We achieved that, and now we want to use it in our crops. We took cucumber. Why cucumber? Cucumber is basically the Arabidopsis of breeding. If it won't work with cucumber, forget about it. So we did this pilot on purpose with cucumber. It works with cucumber. Next, we are doing it with tomato. If that's fine, then we want to use it in all our other crops. Then we have one new genotyping platform that we can use for onion, that we can use for tomato. That's the aim.
Excellent. And what were some of the key findings in this project that your project evaluation found?
Well, we have targeted SNPs. That's why it's called AgriSeq GBS targeted. You know the positions. But you take a stretch of DNA of 200 base pairs and you sequence it. And this 200 base pairs, there might be other variation. That variation is also called. So I got, if you really go into detail, I get 5,000 extra SNPs. Not everybody wants to use them, but if you want, you can use them and you get even more precise information. There is no guarantee because it's targeted GBS. You target 3,000 SNPs, but in return you get 8,000. Yeah, and so then my density is really increased. Yeah, and then I can go, whoa, look, here it is. And then I can work with a small region. And once I have a small region, it's less laborious to transfer it to other genotypes and get the right trait in it.
So the efficiency is...
Yeah, the efficiency will definitely increase.
So what is the overall impact that you expect this to have for breeding programs?
The breeding programs will go faster. We will have more detailed information, so we can, for example, have a better look at relatedness between genotypes, and then we can say, okay, these three are all coming from the same background. We only use one of them because we want to maintain variation, so let's focus on other genotypes which are more different. That's because I have now 8,000 SNPs available. My density, basically my vision is increased. I can look at way more detail. That's an advantage. Breeders, the process will go faster. And that's also a thing, because then I hope that they can come up with the varieties in an earlier stage. So it will definitely have an impact on the breeding program.
What advice do you have for a new lab really kind of starting out to explore this targeted GBS for agricultural breeding?
Well, let's say You should ask yourself the question, where do I want to use the technique for? Is it necessary? Can I do it with a lower density? If that's the case, you don't have to change. But if you think that you want to make progress, go faster, or want more detailed information, then you could consider the AgriSec solution because it, like I said, it has some advantages. But it also, that's the other thing, it comes at a cost.
And what were some of the key findings with the project that you've been working on specifically?
The AgriSec technology is a very sensitive technique. It's based on sequencing, which means that it notices slight contaminations or slight differences. So it's a very sensitive technique, but it's also a highly reliable technique because we did 2 pilots. Well, and the correlation between the 2 pilots was 99%, so better you can't get, which means that they can have repeatable results in time, and that's what we want. We want to always compare, do we make progress, so I need repeatable results.
Well, thank you so much for all the information today, Hans-Peter.
Okay, you're welcome.